Hello students. In this video, we'll learn about the type of lipids. So, in majority, there are two types. One is storage lipids and another is membrane lipids. So, the storage lipids, as the name suggests, are used mainly for energy storage, and membrane lipids are part of cell membrane. Now, storage lipids are non-polar in nature, whereas membrane lipids contain polar parts. Now, storage lipids have glycerol as their backbone, and membrane lipids can have either glycerol or sphingosine as backbone. Now, examples of storage lipids are triacylglycerols, and examples of membrane lipids are phospholipids, glycolipids, and sterols. So, we'll first start with storage lipids, that is triacylglycerols. So, this is the general structure of glycerol, and when we add three fatty acids to glycerol, we get triacylglycerol. Now, this fatty acid can be the same fatty acid on all three carbons of glycerol, or they can be different. And different the use of different fatty acids will give rise to different molecules or different types of triacylglycerols. So this is a simple one. Now moving on to membrane lipids. So the types are phospholipids, glycolipids, and archaeal lipids that are present in plasma membranes of archaea. Now some examples of phospholipids are glycerophospholipids and plasmalogens. Examples of glycolipids are monogalactosyl diacylglycerol, that is MGDG, and cerebrosides. And examples of archaeal lipids are glycerol dialkyl glycerol tetraethers, GDGT. Now let's start with glycerophospholipids. So in glycerophospholipids, one of the fatty acids is replaced with phosphate linked charge group, polar group. Now in glycerophospholipids, the R1, that is the first alkyl chain, is either of 16 or 18 carbon length, and the second alkyl chain is generally of 18 to 20 carbon length, and X is the polar group, and different polar groups will give rise to different phospholipids. And now this is the structure of plasmalogens, where the polar group is choline, and the first alkyl chain contains a double bond. Now, plasmalogens are highly present in hard tissue, in the membranes of halophilic bacteria, ciliated protists, and some invertebrates. Now, moving on to glycolipids, in which first is the galactolipids. Now, in this type of lipids, the third alkyl chain is replaced by galactose. Now, it can be a single galactose molecule, which will give rise to monogalactosyl diacylglycerol, or it can be two galactose residues, which will convert it to digalactosyl diacylglycerol. Here, the first and second alkyl chains come from linoleic acid, which has 18 carbons and two double bonds on delta 9 and delta 12 positions, which means ninth and 12th carbon from the uh, carboxylic acid group. Now, these galactolipids make up to 70 to 80% of the plant lipids. In plants, they are present in the thylakoid membrane of the chloroplast, and because of this high abundance in plants, it makes them most abundant lipids in the biosphere. Moving on to sulfolipids, these are also under glycolipids, where The third alkyl chain is replaced by sulfonated sugar residues. For example, here we have 6-sulfo 6-deoxy alpha D glucopyrenosyl diacylglycerol. Now, in this one also, similar to the last one, first and second alkyl chain come from linoleic acid, and the head group is negatively charged, and they function similar to phospholipids. Now, these sulfolipids are also present in plant, and since phosphate is a growth limiting element for plants, so maybe plants have evolved to use sulfur instead of phosphorus when possible to maintain the negative charge of their cell membranes through sulfolipids instead of phospholipids. Now moving on to sphingolipids. So just like we had glycerol as backbone, in the case of sphingolipids, we have sphingosine as the backbone. Now, when we add a fatty acid to the amine group on the second carbon, and when polar group is hydrogen, we get ceramides. Now, when polar group is phosphatidylcholine or phosphatidylethanolamine, we get sphingomyelins. Now, sphingomyelins derive their name from their location because they are present in the myelin sheath that surround the neuron, and they give the neurons a high conductivity. They help them send the signals faster. Now, what are cerebrosides? So, when we add sugar molecules to the ceramides, we get cerebrosides. The sugar molecules can be either galactose, which the cerebrosides containing galactose are present in the plasma membranes of neural tissues, and cerebrosides containing glucose are generally found in plasma membrane of non-neural tissues. Moving on, we have lipids of archaea membrane. So this is quite a complex structure. The name of the lipid is glycerol dialkyl glycerol tetraethers. We can see here we have the first glycerol, and here is the second glycerol. They are linked by four ether bonds, hence the name with the tetraethers. Now, first glycerol molecule has a sugar moieties attached to the third carbon, and the second glycerol molecule has a glycerol phosphate attached to the first carbon. Now, the alkyl chains in these lipids are very long, 32 carbon long, and these ether bonds are more resistant to breakage at low pH or high temperature compared to ester bonds, and these ether bonds containing lipids may help the bacteria, the archaea, survive the harsh conditions. Now let's look at one question. Given below are few biological lipids in the column X and their chemical nature in column Y. And basically, we have to match them. So, what we have in column X is first sphingomyelin, then archaeal membrane lipid, then C is non-polar lipids, D is plasmalogens, and E is cerebrosides. And in column Y, we have first is galactose linked to ceramide, second is ether linked alkene, third is triacylglycerol, fourth is phosphocholine head group, and fifth is very long branch chain fatty acids. Now let's start matching. So first match, they have given themselves non-polar lipids and triacylglycerols. Now if you look at the options, option 2 and option 3 have C3 combinations. So we just have to figure out a few more. So, as we just discussed, archaeal membrane lipids have very long branch chain fatty acids. So, B5, and there we have our answer, that is option 3. But let's match the rest of them. Cerebrosides are galactose linked to ceramide. Plasmalogens have ether linked alkene on the first carbon of glycerol. And finally, sphingomyelins have phosphocholine head group. So the correct answer is option 3. Now, I know that this is just pure information, like you just have to remember this, the structures and functions, and there is not very much logic behind it to remember it. But as you can see, the this is a question from previous year question papers. So something like this or related to individual lipid functions can be asked again. So watch it once. If you can't remember, watch it again. If you want to read this in detail from reference books, I'll suggest Leninger. It has simple language and it has all the necessary information. Thank you for watching.